Welcome back to my channel. In today's tutorial I will show you how to draw heat map by using GraphPad Prism. Let's start. First you have to click group, and then if you have 3 data then click here. If you have 1 then click 1. I have only 1 data, that means no replication data, so I click here. Next, create. Now I have to import my data here in the spreadsheet. These are my data, so I take 5 gene from different part of the plants: root, seed, stem, flower, etc. And I want to check that which gene expression in which part of plants is higher and which one is lower. To check this, I am creating heat map. So first copy this, all of this gene and their part, and then click here. So these are different parts of plants and these are the genes. So we can click here in graph. So here just click heat map, and then first one is Viridis heat map, and then magma heat map, then inferno heat map. This one is plasma heat map, grayscale, blue scale, and this one is double gradient heat map. I like this color because here the red one is higher and the green one is lower, and the black one is the value near the margin. So I want to choose this, and then I am showing how to change different stuff of your graph. Okay, side by side. So first this one is color mapping. So the range, the largest value, as I told earlier, the largest value is red. If you want, you can change it in for example pink, and then you can apply. So the largest one is, sorry, this is not pink, this one is I guess violet, right? So you can choose this, and if you don't want, then I'm gonna say red is okay for me. Okay, so red is for higher, and the baseline is value black, and smallest value is green. If you want, you can change. So and then graph setting. How can you set your graph? Like page background clear and cell border. If you want, you can keep the cell border, so in every cell there will be border, and I don't need this. And then shape and size, set the size of the cell. If you want to increase the size, you can click here, and others. And if you want to change the row and column, then reverse the order of column, just click here. So it will change the reverse, that is the highest one is here, and from right side to left side, so left side to right side, whatever you want. I want to keep as it is. Okay, and then title, if you want to see the title, just click here and apply. So there's a title, you can click here to change your title name, and also you can change the font size, like 18 forever for example. And then okay, I don't need this. Okay, so then we can talk about the labeling of our genes and different parts of plants. So here in the cell value, if you want, you can put the value of each cell here by clicking just okay. So in each cell there will be the value. I keep it just for better understanding. And then what you can do, row labeling. In our row labeling, we want to change the row label, so just click here, row title, and then apply. So right now the row title is the name of different genes. Now it shows here, we can keep it. And whatever the column title, labeling, we want to see our column titles, so apply. So these are the different parts of plants and their gene expression. And you can also change the position underneath, on top. For example, I need this on top, you can keep it on top, so underneath is okay, right? So, and then the gaps, if you want to place the gap between this cell, you can click here, then apply. See different gap between the vertical gap, and you can also put the horizontal gap. This is a horizontal gap, but I don't need this, so just click here, okay. And then the legend, orientation, vertical overall, that means this one is vertical, you can keep it in horizontal, then it will come here. And you can also change this, like by clicking here to put it up, so vertical is okay. And then location number level, like below, if you want to the numbering right side, you can click here, numbering will be in right side, okay. Left side is okay, okay, right. Then that's all. Then you can click OK, and after that you can export your graph in TF format, download Heatmap, and you can export it, right? So from this heat map we can see that the highest gene expression of ADH1 is found in root, and the lowest expression of ADH1 found in seed, and at the same time the highest PDC1 gene expression was found in root and the lowest in seed. And in case of SOD, highest found in leaf and lowest in seed. That's how we can explain different gene expression in different parts of the plants. Thank you very much for watching.